for the genetic species concept, I went into a little bit more detail about that one. So on the exam, there'll probably be a question regarding some organisms. Let's say, for example, I were to compare like wolves and coyotes and domestic dogs. Wolves, coyotes, and domestic dogs are all within 5% similarity. Only about a 5% difference. 95% similar, 5% difference between wolves, coyotes, and domestic dogs. And they all interbreed and produce fertile offspring. Hybrid wolves, coyotes, called toy wolves in the northeast. Coyotes and dogs can hybridize. So the question there really is more so, what does the genetic differences mean in terms of species? And the answer I've been looking for there was that usually the rule of thumb of species genetic divergence is if there's a 2% difference, that's kind of the rule of thumb for what would make different species. But I was trying to make the point that's based on the human chimpanzee model, that the humans and chimpanzees have a 98% similar DNA and a 2% difference. And that threshold is kind of used as a rule of thumb. The problem is that not all types of organisms, that doesn't apply to all types of organisms. So for example, other mammals might be as much as 5%. And the article that I provided for you about the genetic species concept on Canvas kind of goes into that. But even though 2% is kind of our rule of thumb because of the human chimpanzee model, other mammals might be as much as 5%. Birds might be as much as 10%. Insects can be as much as 15% more. So there's no set, like, if they're this percent difference, they're different species, or the same species. If they're less than that percentage, it kind of varies across all organisms. So that's kind of the answer I want to see on the test, is you to evaluate the percent difference that's presented, and then mention that, well, 2% is kind of the threshold that's generally used, but other species might be as much as 5%. Okay, that's kind of what I'm looking for. In the past, people have been kind of uncomfortable or freaked out about that because there's no right or wrong answer. And that's kind of the point, is the genetic species concept has no right or wrong answer. Right? So I want you to really understand that. And the reason is because right now, the DNA is so easy and inexpensive to sample. It's collected as data because it's kind of impressive looking. But the problem right now is that there's no real answer for how to use that data or what the threshold is, right? So make sure that if you see DNA data, like a percent similarity or difference, that you think about that 2% difference or 98% similarity. It'll probably be listed as a difference. And just note that the 2% is kind of a major threshold point based on human chimpanzees. That's kind of what I want you to know. All right, so that was what we covered up until I got into the fossil record, or how fossils are used to infer macroevolution. Any questions about that stuff so far? Okay. So then we got into the fossil record, and so there will be an example where there's like a mountain or a geological strata image, where I'll ask you questions about what types of rocks would be useful for determining fossil age, and what type of rocks are not useful for determining fossil relative age. This is the relative age of the stratigraphy. And so remember, we talked about two different kinds of rocks: igneous rocks and sedimentary rocks. And I gave you a few examples. Like igneous rocks was lava, ash, pumice, granite. Those are igneous rocks that have come from Earth's mantle. Sedimentary rocks are things like sandstone, shale, mudstone, limestone. I'll give you a few examples. And what you should note, what I've seen a problem in the past, is we're basically looking for the sedimentary rocks. The sedimentary rocks are the only ones that have any usefulness in determining relative age of fossils. Because the sedimentary rocks not only bury the organism remnants, like the organisms die and get buried by sedimentary rocks, but sedimentary rocks have a implied that closer to the surface you are, the younger the rock is, the deeper you are, the older the rock is, right? Age relative presentation. So there'll be questions in there, and make sure that you note or you acknowledge that the sedimentary rock that's further up closer to the surface is younger. The sedimentary rock deeper is younger. So don't miss that one. Fossil. Igneous rocks are useful, but only in potassium-argon dating, right? In potassium-argon dating, you're looking for ash or lava or pumice. Those igneous rock layers are useful for getting a radiometric date, but they are, but usually there's no fossils in them, right? So you're basically trying to get what the actual date is using the potassium-argon dating, and then you're saying that the sedimentary layer is younger or older than that date. That's how it works. I'm not going to ask you to calculate the age of the rock, so this question here is not relevant. You won't have to do any calculations, but what you will have to do is if I give you, say for example, I say potassium-argon dating shows two different dates, 150 million years and 250 million years. You should be able to kind of imply where those come from, like what types of rock, ash, pumice, lava, and then know that the younger date is the one on top, the older date is the one lower down, right? So, but I won't ask you to calculate the age of anything. One question. All right. So, any questions about that? So there'll be like it's like a cliffside. There'll be different layers of rocks labeled, and you'll have to tell me which ones might have fossils and which ones don't. Which ones are potentially older, which ones are younger, and which ones could you apply radiometric dating to? Okay. And then lastly, if I have a question about radiometric dating and ask you about radiocarbon dating, the main answer I'm looking for that one is radiocarbon dating doesn't really apply to fossils. Right? Why is that? What was the main reason I gave for why carbon dating can't really be used for showing that revolution? Does that?
Measure old enough?
Yeah, so it's not old enough. It only goes back, I think the slide I showed said it goes back 30,000 years, but nowadays it's more like 60,000 years because of better equipment. But still, 60,000 years is barely enough to show simple speciation events. So radiocarbon dating just doesn't measure old enough to really be applicable to show the age of fossils. I mean, you can use it for like if you find a woolly mammoth, frozen in the permafrost, in Siberia, that might be applicable. But that's relatively recent stuff, right? So it's not, you couldn't use it for like, Mesozoic determining when arthropods colonized land or something like that. That's way too old. So the main thing I'm trying to get from you on that one is potassium-argon dating is the one that's used for fossil record. Radiocarbon dating is not used for anthropology. It's used for archaeology. So if you want to know how old a pyramid is, you can use radiocarbon dating. That's a couple thousand years old. But or if you want to find out, maybe where the oldest Native American settlement was in the west coast of the U.S., you could, because that might be like 10, 12,000 years old. But anything more than 60,000 years, you can't really use carbon dating, right? But there'll be a question on that. Where is radiocarbon dating potentially useful here? And the best answer is it's not useful. doesn't measure for our purpose, right? Sound good? Just trying to point out questions that people have had struggles in the past with. I thought I was being clear sharing, this. But I think people are not used to hearing the right answer is it's wrong, right? That's kind of what I'm asking you to look for is the right answer is this doesn't apply or it's not. I think it's important for you to know that. Okay, so after that, and then we had this geologic timescale worksheet that I gave you, right? And there's one posted on Canvas as well. So some people have shown me their geologic timescale worksheet, and I've suggested some corrections. If you've done that and want to have me take a look at it, or certainly work together on those, that's fine. You're not turning that one in. I will have one on the exam that looks a lot like that. It'll have the same top part, but the questions will be rearranged and maybe asked slightly differently. So your best bet is to, first off, see if you can answer the questions as written on that page, but then go to the UC Paleontology Museum and try to see if you can come up with maybe one or two more things that happen in each one of those geologic time periods. The older the time period, the fewer things you can list. Right? So the Hadean, for example, only had one thing happen. Nothing happened, right? There was no life present. You can't really ask that any other way, right? But as you get more recent, there are more varied things that can happen. So also be aware of, I don't know, I guess the term red herrings apply, right? So if I ask a question like bats evolved flight, or if I ask the question, whales evolved to the ocean during this time period, or if I ask the question like flowering plants radiate during this time period, that's all the same time period, right? That's all the tertiary, right? That's the time after the Cretaceous, right? All three of those examples apply to that tertiary time period, which is why it's important for you to go take a look at what went on in tertiary, because a whole bunch of different things happen there. But essentially, it was the radiation of mammals. Flowering plants took over, and it'll probably be more animal-based. I was trying to give you examples of. I could kind of spin the question multiple ways. Horses evolved from 3 toes to 1 toe during this time period. Same thing. Yeah. Ben.
So are you saying that there could be somewhere like multiple letters will go in one time period? Or you're just saying
only going to be one letter
okay
for each box. What
but
I'm saying is all
questions.
3 of those questions I suggested referred to mammals evolving into some habitat.
Right.
And that's the tertiary.
Okay.
Even though I could use an example of horses or whales or bats, they're all mammals. So really what the question is, mammals evolved into their habitats during this time period, but using the specific example, I think get confused. I didn't know that. Think back and go, okay, what's that? Okay, that's what the question's all about, right? So anyway, just saying that the questions will be a little different. There will only be one letter per box, so I understand if you get one wrong, you'll automatically have one wrong. One letter goes in one box, right? So we'll work that out. But you can also use that as help, because if you know a letter goes in one box, you put it there, and you know you can't apply it to any other box. Okay, so I'm just saying that because I would like everyone to do that great, to do really well on that. The whole idea was to give you a page that you can kind of knock out of the park and do well on, right? So, but that involves you actually checking it out and practicing. All right, so that's the evolution part of the test. Then we went into physiological constraints. I talked about physiology and we emphasize things like, why do animals need, or animals are heterotrophs, basically they eat biological molecules made by other animals. What types of food resources we talked about? We talked about carbohydrates like sugars and starches, fats and proteins are kind of the main nutritional thing for animals. I think in that regard, the main question there is, among those three, carbohydrates, fats, and proteins, what are the pluses and minuses of each one, right? So, carbohydrates, we talked about that it's kind of fast energy, sugar is readily digestible, great, okay? Fats are great. They have over twice the amount of energy available per gram, right? So fats contain over 2 times the calories per mass unit versus carbohydrates, right? And then proteins are great because you need proteins in your diet to make your own proteins. They have calories similar to carbohydrates, but the problem with proteins is they make a nitrogenous waste, right? You make ammonia whenever you digest proteins, and that has to be dealt with, which we'll come back to. So there'll probably be a little brief compare and contrast question about metabolism of carbohydrates, fats, and proteins, right? And the comparison is they all provide metabolic energy for animals. The contrast is carbohydrates are quick energy, kind of like the reference point. Lipids provide over twice the amount of energy, and then proteins provide energy, but there is a toxic waste that's made when you digest proteins. So that's the idea. Okay, so we talked about feeding, and then along with feeding, there was animals essentially are all aerobic. They demand oxygen in order to break down and metabolize their biological molecules. And in Bio 1 we go into like way detail about this, but here the main thing I'm kind of asking is, why is oxygen necessary for animals to give to us to metabolize their food? And the answer is because they get so much more cell energy with oxygen versus without. I gave you details. They make 32 moles of ATP per mole of glucose with oxygen. They only make 2 ATP per mole of glucose without oxygen. So you could also kind of relate that. That's very close to the actual percent efficiency. They can convert about 30 to 32% of the glucose energy to cell energy with oxygen, and only about 2% without oxygen. So the main thing there is animals don't make enough energy without oxygen to be able to move or do their animal stuff, because animals are so movement oriented. Muscles and neurons, nerve cells, have a huge ATP demand. And without oxygen, animals generally can't supply their muscles and neurons with enough ATP to survive. So as far as I know, virtually every animal is aerobic at some point in their life. And I think I gave you a couple examples of anaerobic animals, right? What were they? Yeah, gut nematodes, they're not moving much, they're just kind of hanging out, right? Feeding on what you feed on. So gut nematodes can be anaerobic. I think I gave you an example of a Nigerian that is a parasite of salmon that's anaerobic. If you Google search examples of anaerobic animals, you could probably count them on your hands, right? Out of the 10 million or so species of animals, there are only like a couple or several that are known to be anaerobic. So I think it's important to kind of understand that and why. But you should note at least that animals need oxygen because they need the extra efficiency, the extra ATP energy release. They can convert about 1/3 of the energy of food into to cell energy with oxygen, but they can only convert about 2% without oxygen. That's really important. Okay, sound good? Okay. Just trying to, just telling you test questions, you know, so hopefully you're kind of getting it here. Okay, and then as far as anaerobic animals, the best one would be like the gun. So that's just so we saw it. Okay, so then I discussed nitrogenous wastes, that when animals digest protein, they make an ammonia waste immediately. And ammonia has some benefits in terms of it takes no energy to make it, and it readily dissolves in water. So it's easy to get rid of if you're a small, aquatic organism. But if you're large and you're not aquatic, you're on land, for example, you can't do ammonia. So I gave you two alternative nitrogenous wastes. And you should know generally what the positives and negatives are and what transport means. So I think I made the point that pretty much animals can, like you make all three. You make ammonia. Your epithelium cells, when they're digesting proteins, you make ammonia. Ammonia just basically evaporates off. Do that. But your muscle cells, for example, will end up making urea. And urea is collected in the blood, concentrated by your kidneys, and then you excrete it by your bladder. So urea is a waste that typically mammals, some other types of organisms use, but vertebrates basically make urea. and then uric acid is kind of on the extreme end of things. Uric acid is nice because it forms a crystal when it's excreted. So organisms that excrete uric acid can get rid of it without losing much or any water. So uric acid is the third type. The problem with uric acid is it requires quite a bit of metabolic energy to make it. So there's an energy investment to make uric acid, but you also don't lose much water. With ammonia, there's no energy, but you need a lot of water to get rid of it. That's kind of the idea. And then urea is sort of in the middle of all three. Organisms that make uric acid, the best one to mention is insects or spiders, and also birds and reptiles need uric acid. But there you go. Any questions on that? Good? Okay, so it'll probably be like a compare and contrast question of the three metabolic states. It'd be important to note energy, toxicity. Ammonia is very toxic, uric acid is relatively non-toxic, and then solubility of water, how much water you need. Okay, then overall, I said, with feeding, respiration, nitrogenous waste excretion, animals have to deal with water balance. So when animals are living in saltwater environments, that's about 3.5% saline. It's a very different experience than when animals are living in freshwater, which might have 0% saline, right? So you should know what it means to be a hypotonic cell versus a hypertonic solution, right? So I went into osmosis. So this is I think this is a relatively straightforward question, but I find in past exams people forget or don't remember, or they do it exactly backwards, exactly opposite of what it's supposed to be. So the easiest way to think about it is the term tonic means soluble stuff, right? Stuff that's dissolved in water, what tonic means. It could be salts, it could be sugars, it could be DNA, proteins. Basically, you can have anything that dissolves in water contributes to the tonicity of water. So tonic means soluble stuff. And hypo means less, and hyper means more, right? Excessive. So if you're given an example of, like, say for example, a fish in the ocean is saltier than a fish, right? So the ocean is hypertonic and the fish is hypotonic, right? So that's kind of hypotonic. So the greater the saline, or the greater the solute, it's hyper. The lesser the solute, it's hypo. And that's where you're remembering. And then the other thing that people mess up or forget about is water moves out of the hypo and into the hyper. So the way I remember that is hypo, O and out, hypo ends with O and out starts with O. So hypotonic loses water. Water goes out of the hypotonic and it goes into the hypertonic. So that's there's going to be a question on that. Probably an example like I gave where there's fish in the ocean versus fish in the freshwater, or maybe an insect, maybe like mosquito larvae in the saltwater habitat versus mosquito larvae in the freshwater habitat. But basically we're looking at which environment is hypertonic, which environment is hypotonic, and where does the water go. So try to review that and try to give you some examples of that. Okay? So those were the physiology constraints. Then I went through how each of the organisms that we've looked at so far, protists, sponges, flatworms, Nigeria, the jellyfish, call it megalodes, how the nematodes, how the segmented worms, and how the mollusks do all these things. So we went all the way up to mollusks. So how I presented it in class was by phylum. We talked about phyla. Some of them had the same adaptation. Like remember I think I said the Nigeria, the jellyfish, and flatworms have basically the same digestion adaptations. They have a gastrovascular cavity. But they had different water balance adaptations, right? The hydra uses their gut to balance water in fresh water. Flatworms had flames and plankton. So how I'm going to do this on the test is I'm going to flip it around. I'm going to say, compare and contrast how, say for example, a flatworm versus a nematode versus a segmented worm, how their adaptations for digestion compare and contrast. So it'll be based on the physiological features. So there'll be a question about digestion, related to digestion or gut tract evolution. There'll be a question related to adaptations for water balance, for maintaining, we talked about osmoconformers versus osmoregulators. And so adaptations for osmoregulation. And there'll be a question about adaptations for respiration. So those are kind of like the main things we talked about was digestion, respiration, and osmoregulation. Probably osmoregulation will be tied to nitrogenous waste, right? So I'll probably tie those two together because they're kind of related, especially once we get to nephridia and that type of structure. We have osmoregulation and nitrogenous waste kind of tied together, right? So, but for some reason, that seems to confuse people because the way I want to ask about it is by physiological constraints. And generally speaking, just to note, I wouldn't really bother asking organisms that have the same thing, right? Like, I wouldn't say compare and contrast digestion of a jellyfish and a flatworm because they're the same. So it would be organisms that have different examples, right? looking for kind of a progression. So sponges had no gut pockets, right? They had to feed on microscopic food that was smaller than their cell. Jellyfish had a gut pocket, they could feed on food that's larger than their cell. And then nematodes had a gut tube where they can ingest food, process it along the way, and excrete the waste out the other end. I think that's kind of the So in that, you're going to have to kind of remember what these violent things are, right? You should know what an body element is or an annelid is. And I find in the past people have forgotten what these things are. That would not be good, right? So make sure that you review your notes on that, because I'm trying to give you the name for it as well as the physiology. Thank you. Yeah.
I think I missed the respiration on the annelida, the second word.
Yeah, the respiration was, they didn't have a specific respiratory structure, but what they did have was a closed circulatory system with hemoglobin to enhance. So they do what's called cuticular respiration. They respire across their skin, but then they have a blood vessel with hemoglobin to carry the oxygen and carry the CO2 out. And then that was contrasted with the mollusks that had a gill of sort of chlamydia, and they also had a respiratory pigment, hemocyanin. So they had similarity, meaning they have a circulatory system where the pigment carries oxygen. The mollusks had a gill, the chlamydia, versus the annelids did not have a gill. That'd be a good one to ask. Compare and contrast the respiration of a flatworm versus an annelid versus a mollusk. And in a flatworm, I mentioned that they basically, their adaptations, they flatten their body so that they have their inner tissues are closer to the outside atmosphere, or the outside oxygen in the water. Whereas the annelid is a larger organism, they have a circulatory system to move the oxygen around and CO2. The mollusk has a gill. That'd be a good contrast to what, right? Sound good? Okay, in that, also to remind you that, well, we'll get there, but I also had questions about open versus closed circulatory systems. So that'll be a question in and of itself. Compare and contrast open versus closed circulation. And I gave you a little. Okay, so any questions about physiology? So just when you're studying, think about maybe write down. You could study maybe by writing down digestion, and then list the organisms out that we talked about so far, protists to mollusks, and then kind of make lists of organisms that have the same adaptation. Protists and sponges kind of have the same, right? They need to feed on microscopic food smaller than cell. Nigeria and flatworms have the same adaptation. Gastrovascular cavity pocket. Nematodes had a gut tube, but it did not have any mesoderm on it, so it was a real simple gut tube. But annelids and mollusks had a gut tube that had mesoderm organs like stomach, crops, cecum, those types of things. So that would be one way to study is to kind of make a list of the organisms and choose the ones that have similar features and separate them from the ones that have different features. That's kind of how the question's going to be answered. And it won't be across all, I won't say, tell me about all the organisms I've talked about for respiration. It'll be specific, like 3 or 4 specific organisms, trying to get you to identify the difference. Okay. Let's see here. Any questions so far? We good? All right. So there will be a question probably in the compare and contrast section, which is like a whole page, asking about the acoelomate body plan versus the pseudocoelomate body plan versus the true coelom body plan. So you should be prepared to kind of describe what it means to have no coelom versus a pseudocoelom versus a true coelom, right? Sound good? Okay. So be ready for that. That was part of the class lecture there and lab. Okay. Any questions there? Also, I've looking for questions about nematodes and their specialized organs for excreting waste. There'll probably be a compare and contrast question that asks you about protonephridia versus pseudonephridia versus trunephridia, right? So I had those organs basically listed for different organisms. The protonephridia was found in flatworms. That was the water pump they used, right? The flame bulbs. Pseudonephridia was the example that was a name I made up, but it's called the excretory tube in the textbook. The nematodes have this unique system that is based on active transport pump because they don't have any cilia. Nematodes and arthropods cells, so they can't use a nephridia to pump wastes out. And then the annelids and mollusks have a ciliated nephridia, which is basically a tube that drains or filters the coelom. It uses cilia to pump coelomic fluid out of the body. So that's what that question's kind of trying to get here is I call it the pseudonephridia, but it's basically the extra of nematodes. Okay, any questions there? All right. I just mentioned this one: pseudocoelimate, acoelimate, trucoelimate, eucoelimate. Okay, nephridia. Compare and contrast of open versus closed circulatory system. There's a couple key features of this, right? So you should, in that example, be able to kind of sketch them both. But you should also know that the open circulatory system had a mixed hemolymph, which is the blood fluid and the coelomic fluid, whereas the closed circulatory system had a separate blood coelomic fluid. So that's kind of a main difference. There's a couple other things that relate to that. Pressure is higher in the closed system, open system low pressure. Energy to maintain. Closed circulatory system requires more energy, open circulatory system requires less energy. Risk to bleeding. Closed circulatory system has a higher risk of bleeding. Open circulatory system has a lower risk of bleeding out. And then you should be able to list the kinds of organisms that have both. At least give you one example. So open circulatory system was mollusks. Closed circulatory system was okay. So is that there'll probably be a separate question on compare and contrast hemoglobin in assignment, but that would also be cool if you mention that open circulatory system tends to use heme assignment, whereas closed tends to use hemoglobin. Okay, and then there's this controversy about the squid's circulatory system. If you Google this, there is all kinds of answers all over the internet, right? And I'm going to tell you that the answers are biased, or they're kind of based on some old textbooks that say that squids have a closed circulatory system. But and again, this is what I find is science students who are learning this have a hard time grasping when something isn't. When the right answer is to say it's wrong, right? And I'm trying to emphasize, I think I've got semesters starting with the giraffes, right? The right answer is it's wrong, is something that you should really begin to develop. And I think this is the most interesting part of science is that you can turn over or falsify an explanation or hypothesis at any time if you have the information. That's the beauty of science. So with the squid circulatory system, even though the textbook, and I bet you ChatGPT would tell you that the squid circulatory system is a closed circulatory system, I'm going to argue it's not. The main reason is they have a mixed hemolymph, right? They have a mixture of their blood fluid and their coelomic fluid. And that's because their circulatory system is not entirely lined with endothelium. They have a gill, their ctenidia, resembles a closed circulatory system with high pressure for efficiency of respiration. But once that blood dumps into the body cavity, it's a hemocoel that has a mixture of both circulatory types. So they basically have an open circulatory system that's modified to function like a closed circulatory system. But fundamentally, if you look at the ancestor of squids, which would be a mollusk ancestor, mollusks have an open circulatory system. So they have adapted their open system to function like a closed system, but they do not have a true closed system. And so that is the answer to the question, right? The answer to the question is, many textbooks and resources say that squids have a closed circulatory system, but they really don't. They've just modified their open system to work like a closed system. And, but for some reason, people have a hard time thinking that way, right? So if you Google this, you will see all kinds of answers. If you use ChatGPT, it'll probably tell you that squids have a closed circulatory system. But you know better, because you know the real facts on this, that squids' ancestors were open systems, and squids have a mixed hemolymph because they have an incomplete endothelium, and so what they really have is a hybrid, right? But fundamentally, it's an open system that resembles a closed system. That's my argument. Okay? This stuff here, dissolved oxygen with water versus terrestrial, we didn't get to yet. So I'm probably going to wait on any of these questions about modification of aquatic systems to terrestrial. The only one we talked about was the mollusks. When mollusks colonize land, what adaptations do they have? And the main thing I was discussing was they have the shell and the mucus, and they also reduce their community size. I think I'm going to go ahead and probably push that out to the next test. And we'll talk about how arthropods colonize land. Because we'll give details on terrestrial oxygen concentration versus aquatic oxygen concentration. Okay, not good? So we'll say that these questions probably don't apply yet. I mean, we don't have enough information to answer these questions very well, so we'll do that for exam 3. Respiratory pigments, there'll be a compare and contrast hemoglobin and hemocyanin, right? So you should know hemoglobin uses iron, hemocyanin uses copper as the metal to carry the oxygen. If you were really good, you'd know that hemoglobin uses 1 iron ion, hemocyanin uses 2 copper ions, right? To carry 1 oxygen. And then you should know that hemoglobin is red when oxidized, hemocyanin is blue when oxidized, and you probably should know the kinds of organisms that have both. Hemoglobin was segmented worms, hemocyanin was mollusks. I gave you a little information about which one's better. It depends. If it's cold and there's not much oxygen, hemocyanin is great. If it's warmer and the oxygen concentrations are elevated, hemoglobin works better. Did I say hemoglobin? Hemocyanin works better cold, low oxygen. Hemoglobin works better warmer, more oxygen. And so it kind of depends on what's going on. Right? Okay. So, I think these questions again are kind of red herring type questions. What end waste might be secreted by an aquatic snail versus a terrestrial snail? The snail is irrelevant, right? The actual snail part is the red herring. It's aquatic organisms tend to excrete ammonia or urea. Terrestrial organisms tend to excrete urea or uric acid, right? That's really what the answer is. I could say snail, I could say worm, I could say vertebrate. That's one thing I'm kind of preparing you for is sometimes in a question there's information that's not necessary for the question, but it confuses people like, oh my gosh, I don't know what snails do. We're talking about animals in general, questions, right? So think about that. Okay, and then we know this one because that's a free. All of this insect, beetle stuff, spiders, we didn't get to. That'll all be good? So, any questions? All right, so that's lecture exams on Thursday. We'll start that at 8, unless you've made other arrangements. And so, if you have any questions during lab today, certainly ask. Right? Good. I left the insect boxes out because I figured that some of you might want to work on your insect key. And so I left those out. But today's lab is going to be a different thing. We're going to look at animal cells and tissues. So we have a number of slides to look at here. So it's kind of a busy lab lecture here. But I will try to tell you specifically the question, the slides that I'm going to ask. But we have to get this animal cells and tissues thing out of the way. And it'll be the last. This will be the last lab topic before our lab. Next week on Tuesday, we'll get the tests back, graded, we'll look at those, and then lab will be able to ask questions about the organization. There's a lot of information in this lab. So make sure you're prepared. All right? Let's take a long, like a 10 minute break, and we'll come back and we'll talk about animal cells and tissues.